My name is Paul Diehl. I'm the COO at Cellecta, and give you a brief introduction to the company and kind of an overview of the technology platforms and products and services that we have. The company was founded about 17 years ago. We're located in Mountain View, California, just south of San Francisco, about a half an hour. And it is a private company. We're not venture funded. We don't have a lot of investors. Our focus, our startup was really based on grants, and from there we developed technologies, and we're still very focused on technology development. We're a small group of about 30 people. And the focus of the technology, the products and services that we develop, are to develop flexible, scalable, and accessible technology for drug target and biomarker discovery. And in particular, to develop approaches for broadly parallel analysis of genes and genetic elements across the genome in a large-scale fashion, but to do so in such a way that doesn't require large-scale automation, LIMS systems, industrial infrastructure, et cetera, so that state-of-the-art labs in cell and molecular biology are able to actually use this technology. And we make the technology as accessible as possible. We typically provide both services and where you can send us samples and have us do some of the work, and product, kits and products for purchase that you can then buy and also run the approaches. And we've been pretty successful with this model. We have over 500 citations that we know about that are on our website. It's a searchable database. If you're interested in some of the technology you hear about, if you're thinking of a study that might that you might pursue, then you can search the database and see if you can find something similar maybe, and that might give you some ideas and also how to do what you're trying to do. And the focus of our platforms are really, there's 4 core technologies that we're focused on. The genetic screening, primarily using CRISPR pooled RNA libraries, also RNAi libraries, which was the way the company was founded on. That was used before the CRISPR revolution, I would say. And these are really to identify the genes that are responsible for different phenotypes that you're looking at. If you're looking at, drug mechanism of action, resistance to particular drugs, just viability of cells under certain conditions, et cetera. These screens allow you to identify the genes that are responsible for actually manifesting that biology. It's different than, say, RNA-seq, where you're looking for genes that are activated or inactivated at a particular point in with a phenotype. And so we also have barcoded libraries. These are sort of an extension of the type of libraries we use for the pooled screening, the genetic screens, and these are for cell tracking and clonal analysis. And I'll talk a bit about both of these technologies briefly, and then, as I mentioned, my colleague, Dr. Chenchak, is going to talk about the targeted RNA expression profiling and immune repertoire profiling, and that'll be the main focus of the talk today. And then also we, related to developing these technologies, have extensive experience and services in genetic engineering of viral constructs and cell lines. So some of you may be familiar with pooled genetic screens. I'll just briefly go over, how they work. But these are really for, as I mentioned, identifying genes that are, say, essential for viability. If you have certain drugs and you're looking to understand why you have resistance of cells to particular drugs that are involved in the mechanism of action, may be critical for biological pathway activation, if you have reporter cells, et cetera. And these are large libraries that are pulled that express sgRNA or shRNA that target hundreds to thousands of genes. And the way it works is we design, say, for CRISPR, sgRNA oligonucleotides that encode sgRNA sequences that target each one of the genes of interest. These are synthesized as a pool, and then they're cloned into a lentiviral library vector, usually some sort of viral vector, but typically it's lentivirus, and then packaged as lentiviral particles so they can be easily introduced into a cell population. And then the number of virus versus the number of cells to introduce them into the population is done in such a way that you get approximately one of the particles into each cell picks up one of the particles. So you can have several cells with the same sgRNA, but each cell picks up one of them, and so it knocks out a particular gene in the cell. And so what you have then is a population of cells, each with different genes all knocked out across the whole population. And now, if you're looking, say, for you do the screen. If you're looking for viability, you can allow the cells to grow for a period of time. If you're looking for activation of a pathway, you can do FACS sorting, et cetera. However you do the approach for the screen, depending on what you're looking for, after the time period, you isolate the genomic DNA from the cells, amplify the guides, and then run next-generation sequencing to count the number of guides in the population. And you compare that with the number of guides, say, in the original library or some sort of control. And the enrichment or depletion of particular guides will tell you about what they do to the cells in that population, and that's presumably because the genes they're targeting are important for the phenotype that you're looking for. And so an example of that shown here, this is a study done by Novartis a few years ago, where they have 5 different genome-wide library with 5 different guides targeting each gene. They made a cell HC, I can't read the number, but anyway, they made a cell line that's resistant to erlatinib. They were interested in the mechanism of action of erlatinib, and they made the cell line express Cas9, introduced the library into the cell line, and then did selection, and then they'd have a treated and untreated arm of the screen. And as I mentioned here, what they're looking for then is they isolate pointers in my. They isolate cells they isolate genomic DNA from each, do next generation sequencing, and then they can look and compare the frequency of particular guides in the population with and without erlatinib. So remember, these cell lines are somewhat resistant to erlatinib already. So when they do this, they find that some of the guides are underrepresented in the treated population versus the MSO population. And so what does that mean? That means that the genes that are being knocked out in these cells make them more sensitive to the erlatinib, and so this might give you some indication of which genes are important for resistance. And then vice versa, they find some that are enriched, and then these targets then make the cells more resistant to erlatinib. And so that means that they may interfere with the mechanism of action in some way. They may be involved in the mechanism of action, or somehow target side pathways that would compensate for the mechanism of action. And so that's a type of screening you can do. This can also be done in vivo. This is a publication from Mirati, who had a compound that was targeting the KRAS pathway and actually targeted RAS itself. And they ran a screen in vivo, and they had done a lot of in vitro screens. And so what they wanted to do was compare, in terms of characterization, the responses that they're getting in vivo and making sure that they're getting a similar mechanism of action for this compound. So they did this with a small library targeting about 400 genes, and then they were able to then correlate that they got similar dependencies, even though somewhat the data looks different because you get different results from in vivo than in vitro, or different responses in some cases. But they were able to then qualify and make sure that the essential genes that were involved in the mechanism of action were also active in vivo. With respect to the CRISPR approach, as most of you know, CRISPR knocks out genes. It uses a nuclease, Cas9 nuclease, that binds to the DNA and then cuts the DNA, and that introduces a mutation into the frameshift mutation in the gene, which knocks it out. But there are newer variations to the system. The lab at UCSF pretty early on had developed a deactivated Cas9 where the nuclease is no longer active. However, it still recognizes the sgRNA and binds to the DNA. And so what you have now is a mechanism to target a specific region of the genomic DNA using an sgRNA that targets that region. And then you can add different activities to the deactivated Cas9, such as in this case, CRAB, which is a transcription repressor, or some sort of transcription activators, and there's a few different varieties of this. And what happens then is you can downregulate or upregulate target genes. And you can also run screens then with libraries like this. So the CRISPR-I, as this is called, CRISPR inhibition of the genes, is similar to the results that you would get with a standard CRISPR knockout. But if you're working, say, if you're looking at long non-coding RNA or some genes that aren't coding, that don't have the standard transcription, then, translation, then, you may not know what to knock out. And so in this case, you can knock down the gene, the whole gene itself, the whole RNA transcript. And then with the transcription activator, the CRISPR-A approach, you can upregulate specific target genes as shown here and get an increase in expression at for the endogenous gene itself. And this can be done on a one-to-one basis shown here. Or again, you can have a library where you're doing a gain-of-function screening, which is a complement to the CRISPR knockout. And we have libraries, genome-wide libraries for all of these different variations of the CRISPR platform, and then we also make a wide range of custom libraries for specialized applications, such as the in vivo screen with the, that I showed you, for example, with the Mirati data. So in terms of making these libraries, we're not just limited to making sgRNA and shRNA. Any short sequences can be made. And so we also have developed a bunch of libraries that have optimized sequences for NGS analysis. They have correct ATCG ratios for sequencing optimally, and they're designed in such a way that there are multiple mutations between each one of the sequences. So you know, you have basically a set of defined barcodes that you can introduce into a population of cells. And with some cloning tricks, you can make millions of different, very defined barcodes that are in a library. And these can be introduced into a population of cells to track the progeny of the cells, to look to create subclonal populations that may have different biology and understand what's going on. In this example from quite a few years ago that we did in collaboration with Amgen, this was one of the first barcode libraries that we made. It was pretty small, about 25,000 barcodes. It was introduced into cells, and then the cells were grafted into a mouse model and then also grown on plates. And then after incubation and a couple weeks of growth, the tumor models were extracted, the cells on the plate were extracted, and the barcodes were sequenced. And what we were able to find out then is from the original barcoded set of cells, how many cells, for example, formed the tumor in the mass of the tumor in the mouse. And as anybody that works with xenograft is probably aware, only a small percentage, in this case it was 6 to 7%, actually formed the mass of the tumor in the mouse of the original founder population. And so this type of study is important to understand sort of the dynamics of the mouse model you might be working with. If you're doing a screen, you need to understand how many cells you actually have to work with to run a screen, such as the one that I showed you for Mirati. The MD Anderson took this approach a little bit farther, and here they use a library with about 50 million barcodes, and they created xenograft mouse, they created cell lines from xenograft mice, PDX cell lines, where they then introduced the barcodes, and they passaged this multiple times, about a dozen times, in order to get a stable population of cells with a number of different barcoded subpopulations, thousands of different barcoded subpopulations. And they could use this to look at various drugs and how various drugs affected the cells in terms of killing off certain sets of cells. Did two different drugs kill off the same barcodes, for example? If they did, that would indicate similar resistance. Were certain cells in the population already primed to be resistant to particular drugs, or is that something that evolved after the treatment, et cetera? So they were able to do that, and they were also able to do that in mice, similarly. This is a little bit different configuration for the barcodes. This is set up at the end of the transcript where the barcode is, and so this is at the 3' end of the transcript where it can be detected if you're using single-cell RNA sequencing. And in this case, Genentech used this approach to look at a couple different analogs of erlotinib. And what they did was introduce one that allows degradation of the tyrosine kinase inhibitor that the erlotinib binds to, and one that inhibits degradation of that, protein degradation of that. So both of them inhibit the tyrosine kinase, but one of them prevents also degradation of that. And they were able to introduce that into the cells. They ran the 10x single cell system with 3 different treatments. And so the point here, though, is that when you treat the cells, what happened is the cells that are missing are the ones that have maybe the interesting biology. Why were they susceptible to the drugs, and these ones are resistant? And you can look for specific differences, because in this one you can go back and look at the cells here before treatment that you still have those cells, and you have the data from what their expression profile is, and you can compare that to the ones that were susceptible, and then you can get a direct comparison. And so in this case, what they did is they did that sort of study, and they found that the cells that where the inhibitor did not prevent degradation, were less susceptible to the drug. And when they looked at the data and compared the expression profiling of susceptibility to the various drugs to the expression profile, they were able to see that there was actually a much lower expression of endoplasmic reticulum-related genes, and the ER pathway was not as active in the cells that were susceptible to the one that allowed degradation. And so that indicates that there's some indication for some need for the degradation. It contributes to the toxicity of the erlatinib. And so then finally, I won't go into a lot of detail here, but you can combine barcodes with RNA libraries. In this case, we were working with a group that has had sgRNA to several different tumor suppressors, and so you knock out the tumor suppressor, you probably get a tumor. These were combined, each of these with where they were also labeled with thousands of different, hundreds of thousands of different barcodes. And so each sgRNA and barcode combination is unique in the library. And then that was introduced into the mouse. This is a respiration model, goes into the lungs, it infects the lungs. The lungs can be isolated, you can do next generation sequencing, and basically looking at the number of barcodes for a particular sgRNA, you can see how many cells roughly are infected in the lungs in those animals. And then the number of counts that you get for a specific sgRNA barcode population will tell you the size of the tumor that was formed. And so you can then do a profile and see the size of the tumor that was formed with each one of these knockouts and the number versus the number of infections, basically how tumorigenic they are. And then, of course, they went and treated with different drugs and see how that affects this profiling.